Hello and welcome. My name is Victoria Rees and I'm the Deputy Editor of Drug Target Review. I'm joined by Ann Ager, Professor of Cellular Immunity and Immunotherapy, Division of Infection and Immunity and Systems Immunity Research Institute in the School of Medicine at Cardiff University. Ann will be speaking at Drug Target Review Cell and Gene Therapy Advancements online summits. Click the link below to reserve your spot and hear about Professor Ager's latest research. Today, Professor Ager will be discussing her research into CAR T cells against solid tumours. But first, let's get to know her. So, Anne, would you be able to give a brief background to yourself?
Yes, so I just want to say thank you very much, Victoria, for this interview and opportunity to give a preview and highlight what's happening in the CAR T cell therapy field. So I am a cellular immunologist. So I'm an academic researcher currently based at Cardiff University, but I've had, I guess what you might say, a conventional academic career after my PhD. I trained actually in blood vessel biology, understanding how blood vessels have to keep healthy in order to keep all our organs around the body healthy. And then after my postdoctoral training, I moved into immunology and started to work on really how the immune system is organized in our bodies and how immune cells have to move around the body and move to where infections are, like virus infections. I mean, we all know about COVID-19. We've learned so much over the last year that the virus replicates in the lungs, but our killer T cells and other immune cells need to home to the lungs in order to clear the virus. So that's the sort of research that we do. We use different models. We use various types of preclinical models, both cellular models and animal-based models. And the idea is to understand how we can target T cells, particularly killer T cells that will kill cancers, to solid tumours. And this is, I guess, why I'm going to be talking to you about our work. Yes. Oh yes, and of course I don't do this on my own. I have a great team. I've been very fortunate throughout my career to always very pleased when people want to come and work on the things that interest me. So I have a team of young PhD students and postdoctoral fellows. So these are people who've just finished their PhD, who are starting their careers in research and research assistants. And we work very much as a team. So the last year has been very difficult because normally we'd meet in a room and look at our data, the results that we've got, and try and troubleshoot if things aren't working and plan future experiments. And I have to say that's one thing I found very hard doing on Zoom. So I'm really looking forward to being able to get back to face-to-face meetings as soon as is safe, of course. Yeah.
Definitely, I think everyone can empathise with you there. Yeah. Fantastic, thank you so much. So I first wanted to ask you more generally about your research, but why do you think it is important to develop new therapeutic strategies against cancer?
Well, of course, the figures that you can find on every national cancer charity's website tell it all. So, if you in the UK, Cancer Research UK is really the leading national charity, and there are 1,000 cases of cancers, any cancer, diagnosed every day, and 450 deaths a day in the UK. Obviously the leading cancers, as it were, the most frequent in sort of lung and breast cancer and colorectal cancer, get a lot of attention, and that's understandable. But of course, there are many other types of cancer, which I guess that if an individual is unfortunate to have perhaps develop one of the rarer cancers, they feel they're not, they're not actually perhaps being looked at or being catered for, as it were. So obviously, cancer is a dysregulation of tissue cellular growth. So there are many drugs that have been developed, are being developed to target that particular aspect of the cancer cell, how to stop the cancer cell dividing rapidly and taking over and depleting our bodies of what our bodies need. So there are many drugs that have been developed, and I guess that the major advance over the last few years has been able to target drugs, being able to do molecular mapping of what the genetic changes are in a cancer, to be able to say, well, this cancer has this mutation, therefore this drug is going to work better. So we do have an armament, I guess, a good armory of drugs that target the cancer itself. But of course, we know that cancers develop, it's Darwinian evolution, cancers evolve and will evolve to be resist the drugs that are being used. And it's a battle, it's a constant battle. I guess the major breakthrough, again, in the last sort of 10 years or so is the realization that the immune system is actually in battle with the cancer. And we've known for many, many years that immune cells, we can find them inside cancers. Histopathologists looking at resected cancers or biopsies have noted immune cells for many years, but we haven't really understood what they might be doing there, whether they're cause or effect type thing. So I guess the breakthrough really has been in the last sort of 8 to 10 years, realizing that actually we can harness the patient's own immune system to attack the patient's own cancer.
So in terms of cell therapies, and in particular CAR T cell therapy, what kind of advantages could they provide when tackling cancer?
So the CAR T cell therapies are particularly attractive because the issue with targeting the immune cells to attack cancer is obviously you have to have a specific receptor that is specific for the cancer and not for the patient, normal healthy cells. So you don't want your immune system to be attacking your normal tissue. Otherwise, you get autoimmunity, and we know autoimmune diseases exist. So we know how powerful the immune system can be, both antibodies and T cells. Both CD4 and CD8 T cells can be very underlie autoimmune conditions. So we know how powerful these T cells are. So the beauty or the attraction of CAR T cells is that what the cancer specific receptor on those cells is not the natural T cell receptor. It's a chimeric or a hybrid molecule. So the outside of it is actually the same as an antibody. It's the FAB binding part, the antigen binding part of an antibody, and then it's hooked up to the transmembrane and all the signaling properties of a T cell receptor. So it's very much a B T cell receptor, a hybrid receptor. And the attraction of that is that you no longer rely on the conventional T cell receptor recognition system, which is very much peptide derived from inside a cell being presented on the transplantation or HLA antigens on the cell surface. So this bypasses that, and so it gives you much more broad specificity. It also gives you potentially higher affinity. We know that antibodies are a much higher affinity for the antigens. And you've got then the opportunity of having a synchronized attack, because all of your T cells have the same T cell receptor or the same CAR receptor. We know that obviously the success of CAR T cell therapy has been amazing against blood cancers, leukemias, but just to explain that in a bit more detail, the antigen that's been used there has been very much CD19, the CD19 antigen or CD9 molecule, which is on all B cells, including B cell derived leukemias. So CAR's receptors have been developed to target this CD19 antigen. So basically you get a killing of the leukemic cells. You also take out some normal B cells, but because B cells are generated every day from the bone marrow, we know that there's a recovery period, so that's the hit, that's the what's called it off-target effects that you have, collateral damage that you take with that therapy. But patients survive survive. And we know that young patients do very well. And one thing we think that the younger patients perhaps do better than the adult patients and older patients is obviously cancer. Solid cancers tend to be a disease of older age, and we know our immune systems age as well as our body's age. It's just part of the body. So it may well be that the CAR T-cell therapies have shown some very, very stunning successes in younger patients because their T-cells are younger and fitter and healthier. So if we can understand why that is, maybe we can translate those properties of young T-cells onto like my T cells. My T cells are older, make them better. So that's great for leukemias, and that's been the breakthrough. Now, targeting solid cancers is much more difficult. So first of all, it's finding an antigen on the surface that's specific for the cancer and not on our own healthy cells. That's been very difficult. And also every cancer will have a different set of antigens on the surface that you could potentially target. But then the issue again also is that the antigens could evolve. The cancer cells will mutate, continue mutation, and change their antigens, so may no longer be a target for the CAR that you choose. So that has been difficult. The other thing that's difficult is that we know with the therapy for leukemia, the CAR T cells are engineered obviously outside the body. Patients' T cells are collected from their blood and then genetically modified outside the body and then infused back in. They're infused into the bloodstream, and that's exactly the same compartment of the body that the cancer is in the case of leukemia. So we know there's no issue of trying to get the T cells to meet up with their target cancer. Whereas for solid tumours, infusing CAR T cells back into the patient's bloodstream, the tumours are not in the bloodstream. So those T cells need to know where to go in the body. They need to traffic to where the cancers are. And that's very much something that, the is great interest to us. So that's one of the hurdles, but there's the possibility that you could inject CAR T cells directly into cancers, particularly if the cancers are external, like head and neck cancer, and perhaps even for primary melanomas.
Could you just outline your own research into CAR T cells that could potentially be used against solid tumours?
Yeah, so as I said, introduced, we are interested in how T cells move around the body and how they target tissues in order to be effective. We work on virus clearance, and actually we made a breakthrough in our studies of virus clearance. We found that a particular molecule that's known to be important for T cell homing on lymphocytes, we were looking at it on CD8 killer T cells. This molecule is normally downregulated or lost from the surface of T cells when T cells become activated. So the processes that T cells go through to be engineered with a CAR, which involve proliferation and lentiviral transduction and expansion to generate large numbers of cells to infuse back to patients, those processes actually induce loss of this homing molecule. We know that, and that's been in the literature. It's been known for many years. So what we've done is engineered a gain of function form of this molecule. It's called L-selectin or CD62L. And so what we have engineered is T cells which basically don't downregulate L-selectin when they're activated either in vitro and go through proliferation cycles in vitro, or in experimental animal models. We use preclinical mouse models of cancer immunotherapy. So we've set up an adoptive T cell transfer model, very much like a CAR T cell model, although we're actually not using a CAR at the moment. We've used a cancer-specific alpha-beta T cell receptor, but the principles are the same. So we've engineered the T cells to express a cancer-specific receptor, and they express this gain-of-function L-selectin. And when we use those T cells for adoptive T cell therapy, injecting them into the bloodstream of tumor-bearing mice, and these tumors are growing subcutaneously, we've shown that these T cells are better at controlling tumor growth. So we see more killing of cancer cells, and we see more T cells getting to the solid tumours in these models. So we think it's our hypothesis was this homing molecule, by maintaining at the surface, would offer a homing advantage, and that's what we see. More T cells get there, and we have better control of tumour growth. So we're currently now testing it alongside a CAR. So we're engineering, and now we're working in the mouse, a preclinical model. We're now moving into human cells, as again in doing ex vivo preclinical studies, and asking, can we repeat this in a human T cell, but using a CAR instead of an alpha beta T cell receptor. And there are many several potentially clinically relevant CARs for solid tumours out there that we could test. So that's what we're doing. But the other point I really want to make, and I'm quite passionate about, is that not only engineering the T cells so that they home to solid tumours. So that means that when they're injected into the bloodstream, they're then recruited out of the bloodstream into the cancer microenvironment in order to kill cancer cells. You've also got to think about the tumour blood vessels, because without blood flowing through the tumour, you wouldn't get any delivery of CAR T cells, you wouldn't get any delivery of cancer therapeutics at all. So the blood flow to the tumour is very important. And we know that tumours or cancers, just like mutating to hide from the immune system, they actually disrupt the blood vessel organization in tumours and or cancers in patients. And there are blood vessel targeting molecules. So there's molecules such as anti-VEGF, so vascular endothelial growth factor, which has been approved for colorectal cancer. So what these blood vessel targeting agents do is tend to mature the tumour blood vessels, so you get better blood flow, better oxygenation, better delivery of nutrients and clearance of metabolites. And so what we're trying to do is combine the T cell therapy with the vascular targeting agents, the blood vessel normalizing agents. And certainly in preclinical models, this combinatorial approach is very powerful. So I think relying on one therapy, I think, is not going to be enough. I think the advantages are going to be in thinking about hypothesis-driven combined approaches. Think about which pathways you're targeting and whether they're going to be additive or really synergistic, which is really what we want. Yeah.
Well, thank you so much for speaking with me today, Professor Ann Ager, and for giving such excellent answers. It's been an absolute pleasure.
Well, thank you, Victoria, and thank you and Drug Target Review for organising this meeting. I'm very much looking forward to the session. Obviously, I've spoken about our passionate interest and our ongoing research. There are many other aspects to CAR T cell therapy and also homing. I didn't mention chemokines at all, but I'm sure they will be covered. And all I can, what I'd like to say is really do feel free to get in touch with any questions. And if I can't answer your question, I think I might be able to point you in the direction of someone who can. So thank you very much for listening.
Yes, and thank you for watching. And if you would like to hear more about Professor Ann Ager's research, do join Drug Target Review's Cell and Gene Therapy Advancements Online Summit, taking place on the 3rd to 4th of November this year. If you sign up using the link below, you can learn more about targeting CAR T cells towards solid tumours. We hope to see you again soon. Thanks for watching. Goodbye.